And now what we need to do is go to that sample genetic elements.dat file, turn it into a genetic elements.dat file, and edit it to point to the genetic elements we just downloaded. And don't worry, it's nowhere near as complicated or annoying as that makes it sound. So let's go take a look at that file. Okay, so I've gone ahead and opened the sample genetic elements.dat file, which was living in the same input directory that we just stuck those 4 files in the WordPad editor on my Windows machine. I recommend using the simplest editor you can for this, just so your WYSIWYG, complex, nifty text editor doesn't accidentally add in a bunch of invisible tags that will be really visible to our software and will cause problems. So we have a full set of directions here telling you everything you need to know. I'm not going to linger on them, but they're there. Read through them when you're actually doing this yourself. It tells you what each little thing is for. So a quick review: ID, you're just identifying the genetic element. A name, it's a descriptive name. The type, the 2 types we're going to care about right now are chromosome and plasmid. And the default condition is chromosome, whether it's circular or not. What codon table to use. So for example, if you were doing a eukaryotic organism and you would set the general codon table to whatever it is, but you wanted the mitochondria to naturally have the mitochondrial codon table, you can do that. A link to the annotation file, that's the GenBank file. A link to the sequence file, that's the FASTA file. You can get away without having this, but you might as well have it. And the contig file if you're actually using contigs and not, say, chromosomes and things like that. So let's scroll down. And we have a bunch of test things filled out ahead of time depending on what kind of conditions you might have. I'm just going to delete the ones I don't need right now, but again, I suggest that you just look through everything when you're working on this. Then I will copy the one that I'm going to use here and duplicate it. And now let's go ID. So the first thing is going to be just our chromosome, and we'll just call it chromosome because we just have the one. Name, we'll just go with chromosome. The type is indeed chromosome. Let's change this because it's circular. And now we went with Chrome1 as the name here, even when I saved them. But of course it's not a PF file. And if you're wondering what PF is our sort of homemade annotation format, pathologic format. And that's what we actually convert things into when we do our big BioCyc builds where we're doing 100 things at a time, because it lets us sort of pre-filter to make sure that we can actually parse all the stuff we're working with. But we don't need to worry about that now, and you don't need to worry about that when you're working at home. So it's just the GBK. And then FSA is one possible suffix for a FASTA file, but in our case they're called FNAs. Then our second genetic element is our plasmid. We just have the one again. And we can change the name to the evocative name plasmid. And the type is obviously not chromosome. It is, in fact, according to the comments above, plasmid. Now, if you're wondering where I got that change from, let's click up here. And you can see under type, types are chromosome with this sort of chromosome thing, plasmid, and MT for mitochondrial chromosome. So it's a plasmid. Yes, it is also circular. Its name for the annotation file is plasmid.gbk. The sequence file is plasmid.fna. Okay, so we have the information we need to point the software to the right places to look. So now let's save this, but we don't want to really save it into the sample genetic elements. We need to save it as something else. So save as just geneticelements.dat. And we're good with it just being a text document. We save. So now we have a geneticelements.dat. We can get back to the process of actually making our database. Okay, so we're set. We have our files. We have our geneticelements.dat file. What do we do now? Well, at the bottom of the page, the software is telling us we can either do a trial parse or we can go directly to the build. Might as well do a trial parse. It lets you know if you've made a general mistake that will prevent an actual build. So we go up to Build, click on that, Trial Parse. It'll ask you if you want to do any one of your genetic elements or all of them. Let's just select all and hit parse. Aha! So we have a problem. Now if you were watching carefully at home, you'll notice that I left something out when I was saving the file names, because even though we just have the one chromosome and the one plasmid, I numbered them both. Oh no, a mistake. This is why trial parse is handy, because it's not going to go through a bunch of work and then spit up at you. It's going to spit up at you right away that there's a problem. So let's dismiss this, and I'll go edit that mistake and then come right back. Okay. Okay, that example is admittedly a little cheesy, but that's one of the values of trial parsing. It tells you if there's a problem. And you can see while I was away editing my genetic elements.dat file, it actually went through, parsed what it could, told us, hey, I found some genes, I found some proteins, I found some RNAs, and I didn't find any of that plasmid stuff you were talking about. So once you edit your genetic elements.dat file to get it to work, you'll need to get out of the trial parse and then try it over again. So let's try that over again. Build, trial parse. Let's just do the plasmid, parse that. Okay, trial parse is done, and it told us, why yes, I found 67 genes that make 67 proteins on your plasmid. And one of them was matched with the enzyme name matcher. So not a lot of enzymes on the plasmid, but a lot of proteins. Great, so now we're ready to actually build our database. And what building entails is going through the GenBank files and pulling out of that gene names, gene products, functional information, adding enzyme reactions on, all that stuff that I talked about before, and layering it onto that basic skeleton that we made at the very beginning. Okay, so let's do that now. Build, automated build, and it's off. And this is the part where it's not going to take that long, but you could certainly walk away from the computer if you wanted to, or you can sit here and stare at the screen as it tells you that it's found some ambiguous EC numbers that it doesn't feel like trying to match, and that it's now matching enzyme names. In general, I don't sit around and watch these reports, and you don't have to either. It's actually logging all this, so you could go back and take a look at it if you really cared. Of course, you'll just see the results in the final product. And you'll notice here it talks about pruning out variant or likely false positive pathways. And what that means is that, as I mentioned, we don't want to overpredict too much. So in this case, say variant pathways. If you look in BioCyc, we may have 5 different versions of glycolysis, for example. We don't really think your organism is going to have all 5, but there's a good chance there's heavy overlap between the different reactions in the different glycolysis pathways. So we predicted all 5, say, or all 3, or all 2, and then we're going to pick the best we can, the best 1, or maybe 1 or 2, out of that set and dump the rest. Similarly, there are other ways we have to detect likely false positive. These are discussed in the publications. Okay, so now we're going to let it update the overview graph. That can take a little while, though not usually too long, and we'll come back when it's done.